My name is Michael. I work at Epic, and my question is, how can we use stem cells to build tissues and organs?
My name is Dave Verady, and I'm a fellow at the Morgridge Institute for Research. And my area of interest is regenerative medicine. There are two major properties of a stem cell, and the first one is its potency. And by that we mean its ability to give rise to specialized cells. There's this hierarchy in stem cells depending on how potent they are. And at the very top are the pluripotent stem cells, which can give rise to everything. The second component to a stem cell is this term called self-renewal. And by that we mean the ability of a stem cell to regenerate itself. All of us walk around with stem cells that sort of repopulate our blood and our muscle tissue and our skin tissue, and we're in a constant state of renewal. To get back to the question of how do we make tissues and organs from stem cells, the trick is to understand the signals that tell a stem cell how to give rise to all the different cell types within that tissue. You can imagine that that's a lot of trial and error, because there's no sort of manual that tells us how to go from a stem cell to a particular tissue, or how to go from a stem cell to a particular organ. The biggest challenge right now is that we can push a stem cell to a single cell fate or specialized cell state. But in a tissue, there's multiple cells. An organ like your heart or your brain is not just made up of one cell type, it's made of many cell types working together. How we structure an organ to have all that cellular diversity is still largely unknown. So learning how to tweak the system so you get just the right ratios of these different cell types that then engage together to form a really functional and useful tissue, I think will be a key next step.